Hi everyone, my name is Wen-Bing Jin. I'll introduce the paper submitted to iMeta. The title is Genetic Manipulations of Non-Model Gut Microbes. Human microbiota means all the microbes living on or inside human body. As shown in this picture, several body sites including nose, mouth, skin and gut are enriched in these microbiota. It has been reported that the total number of these microbiota, especially bacteria, are 10 times more than our total human cells. We know that gut microbiota is also known as invisible organ, which plays important role within host, such as digestion of diet and regulation of immunity. Dysbiosis of gut microbiota cause a series of host diseases, such as IBD and colon cancer. Multiomics show that gut microbiota genes are closely related to host biology, and we focus on how gut microbiota genes control the production of metabolites and then has an impact on host biology. To explore the functions of genes which are mostly encoded by non-model gut microbiota, genetic tools for those non-model gut microbiota is necessary. Here, we are introducing a step-by-step genetic strategy that facilitates the study of functional genes encoded by gut commensals. This strategy can help us to establish genetic tools in non-model gut Bacteroidia and Clostridia without prior knowledge of genomic sequence, and can serve as a high-throughput genetic screening and manipulating platform for human gut microbes. For Bacteroidia microbes, we first screened the culture condition and antibiotic resistance, and we selected 16th rRNA as a common target gene. Then we use single crossover integration to evaluate the genetic tractability. After this, we were able to knock out other target genes of interest. For example, we knocked out asparaginase gene in one Bacteroid strain and verified the gene knockout by diagnostic PCR and LC-MS. Further, based on this, we developed double crossover deletion tools and knocked out gene TDK in another Bacteroid strain. For Clostridia microbes, we firstly screened the culture condition and antibiotic resistance, and developed a mixed conjugation strategy to efficiently identify the compatible replication origin, which enable the plasmid to replicate in the recipient strains. The compatible replication origin was confirmed by diagnostic PCR and sequencing, and verified by single-strain conjugation. Based on this, CRISPR-DCPF1 system was selected, and assembled into the plasmid containing the compatible replication origin together with the report gene LAX to serve as the control group. Then gRNA locus targeting the promoter encoding sequence of LAX was introduced to serve as the mutant group. After these two plasmids were transferred into Clostridia microbes, qPCR was performed to quantify the downregulation of expression of gene LAX. Besides, we also developed Group 2 intron system for Clostridia microbes. From the reported starting plasmid, we replaced the target intron with the 16th intron that target the common 16th rRNA gene to get a set of plasmids PGMXCAQ. Then the retrotransposition activated markers was changed with AAD9 to generate another set of plasmids PGMXCBQ. These plasmid was transferred into the recipient Clostridia microbes and successful gene insertion was selected by RAM antibiotics, diagnostic PCR and sequencing. In summary, in this protocol, we introduced an efficient and generalizable approach to establish genetic tools in non-model gut microbes. For Bacteroidia microbes, genetic tractability was screened using single crossover integration, and gene knockout could be performed via single crossover insertion and double crossover deletion developed based on this. For Clostridia microbes, plasmids with compatible replication origin were firstly identified, then CRISPR-DCPF1 mediated suppression and Group 2 intron-induced insertion genetic systems were developed.